This is chapter 18, which is the cardiovascular system, and specifically we're just looking at the blood and the components of the blood. So what makes up the blood? Well, you've got the fluid portion, which is the plasma, and then you have the formed elements. Blood is a connective tissue, and remember, with connective tissue you have your cells, and then you have that extracellular matrix. Well, blood is the only one when that extracellular matrix, instead of being solid, is a fluid, and that's the plasma. And so the cells are the formed elements. You have the red blood cells, which are known as erythrocytes. You have the white blood cells that are your leukocytes, and then the platelets, which are known as the thrombocytes. Overall, the function of the blood, number one, is transportation. You have nutrients that are dissolved in the blood. You have oxygen, carbon dioxide, other waste products. You have hormones. All of these are being transported from one area of the body to another area where it's needed, or like waste products where you have to get rid of it. So that's part of the function of the blood, is to transport all of these items. The white blood cells are involved with defense, so your blood does play a role in defense because of the white blood cells. And then maintenance of homeostasis. Homeostasis, remember that internal balance, you're maintaining temperature. Well, blood does carry heat with it, and so it can play a role in helping to maintain your temperature. In terms of chemicals, different ion concentrations, whether it's sodium or calcium, a lot of that, the concentration is monitored in the blood. And so there's a range it can be at. If it gets too high or too low, adjustments will be made, but it's going to be monitored in the blood. Same thing for pH of the body. It's actually, the pH is monitored in the blood. And then the water content, in terms of trying to keep you from getting dehydrated. So what's the composition of blood? Well, you have the plasma, the fluid. That is composed of water. There's proteins in there. There's nutrients in there. There are hormones in there. There's what we call the Buffy coat. When you centrifuge sample of blood out, it's going to separate into these three different layers. It has to do with density. The Buffy coat is going to contain your leukocytes and your thrombocytes. Remember, those are your white blood cells and your platelets. And then the hematocrit, that's going to contain the erythrocytes, or the red blood cells. So when you take a sample of blood and you spin it out, the red portion down here, because it has red blood cells, that's your hematocrit. That Buffy coat is this thin layer in between the hematocrit and the plasma. If a person is anemic, they have fewer red blood cells, so you're going to have less here. And then if they have polycythemia, it's too many red blood cells. Just some general characteristics of the blood. The color, when it is oxygenated, has a lot of oxygen, it's bright red. It's a darker red when there is less oxygen in it. It's viscous, meaning it's kind of sticky. And we'll see towards the end of this chapter, if you have too many red blood cells, the blood actually can get thicker and viscous. Think of like syrup. And so it's not going to flow as freely as it should, and that's oftentimes why people be placed on blood thinners to try to make so it flows more freely. The temperature is about 38 degrees C, so it's a little bit warmer. Internal body temperature is usually on average about 37, so the blood itself is a little bit warmer. As I said, it carries heat with it. The pH on average is 7.4. The pH of the blood must be maintained between a pH of 7.35 and 7.45. If it drops below 7.35, you can start having problems. If it goes above 7.45, you can also have serious problems. How much blood do you have? Well, males on average have between 4 and 6 liters, and females, it's a little bit less, between 4 and 5 liters. We look at the blood plasma, the proteins that are often found in the plasma under normal conditions. One of the ones is albumin, which is produced by the liver. It does help to maintain your osmotic concentrations. It helps to transport various lipid molecules. You have globulins. These are involved with immune responses. Some of them are involved with transport and also maintaining osmotic conditions. And then you have fibrinogen. This is necessary for blood clotting. The solutes, the things that are dissolved in the fluid, are number one, your electrolytes, things like sodium, potassium, and calcium. And then oxygen and carbon dioxide, nitrogen, some of your organic nutrients, your vitamins, glucose, which is sugar, amino acids, and waste products are also going to be uh, classified as solute. A solute is anything that's in the solution. And so this table is just showing you the division between the plasma and the formed elements, the cells, remember, and then this is breaking it down percentage-wise, where you can see most of what's in the plasma is water, and then here it shows the major proteins and then some of the other solutes, and just what their overall function is. And then we're going to be talking about the formed elements in a moment as well. The formed elements, if you break those down even further, and this is where we're going to talk about each one of these separately. You have your red blood cells or erythrocytes. You have your leukocytes. There's 5 types or classifications of these, and then the platelets. The production of the formed elements, all the 3 types of them, the platelets, white blood cells and red blood cells, they are produced usually in the red marrow. They initially come from stem cells, and then differentiation will occur. Differentiation just refers to when they become specialized, so that they all start from stem cells, and then they receive these, it's kind of like receiving a message that, okay, you need to form this type of cell. Well, you need to become this type. That differentiation is when they specialize. So hemopoiesis is that process of forming the formed elements. There are going to be different growth factors involved. Erythropoietin, or EPO, is secreted by the kidneys. It is secreted in response to low oxygen, and the result is that you're going to see an increased production of erythrocytes. And why is that? Because oxygen binds to the red blood cells. So if you have low oxygen, how are we going to get that oxygen level up? Well, the oxygen needs something to bind to, so increase the red blood cells. Thrombopoietin is also produced by the liver and the kidneys, and this is going to increase the platelets. Cytokines, these will stimulate both what we call specific and non-specific resistance to diseases. So let's look at the erythrocytes, those red blood cells. In terms of their size, they're about 7 to 8 micrometers, so they're very small. And just so you know, in A&P and often in like a general biology, we tend to call this unit micrometers. 1 millimeter has 1,000 micrometers in it. So micrometer and millimeter are not the same thing. This is much smaller. Like I just said, there's 1,000 of these in 1 millimeter. In microbiology, we often use the term micron. A micron is a micrometer. So I sometimes switch because I'm more used to calling it a micron. So if I use it interchangeably, it's the same thing. Okay. They're small. Their purpose is to transport the oxygen and the carbon dioxide. The erythrocyte in the differentiation stage, it starts as a stem cell, and then, like I said, it goes through differentiation, and as it goes to mature, it actually will lose its nucleus and most of the organelles, including mitochondria. That way, its pretty much function is to carry that mostly oxygen. You can see the shape of it. It's biconcave. That increases surface area. It also makes a little more. It's very flexible. It can kind of fold in on itself to get through some of the blood vessels, like the capillaries that are very small diameter. Only about one cell can get through at a time. And sometimes they have to kind of like fold over on themselves to pass through. In the red blood cell, in the erythrocyte, there is hemoglobin. This is a protein. There's actually thousands of these in each cell. The protein itself is made up of 4 subunits. We refer to the subunits as alpha and beta, so there's 2 alpha and 2 beta subunits. Each one of the subunits has an iron associated with it. And why is it important to know that there's iron associated? It's because the iron itself is what binds the oxygen. So that means per hemoglobin molecule, or protein, for each one of these proteins, if it's fully saturated, it can bind 4 oxygens. Oxyhemoglobin just means that the hemoglobin has oxygen bound to it, and it's a very bright red color. Deoxyhemoglobin is when there is no oxygen bound, so it's a very darker red. And then when carbon dioxide is bound to it, because sometimes it'll drop off the oxygen in the tissues where it's needed, and carbon dioxide is a waste product from the cell. So we'll pick up the carbon dioxide, transport it back to the lungs where you exhale it. When hemoglobin has carbon dioxide bound to it, then it's known as the carbaminohemoglobin. And so this is what it looks like. You've got these subunits. So down here on the bottom, these 2 are your alpha subunits, and the 2 on top are the beta subunits. Just know that there's 4 subunits, and right here, this is showing the iron. So in here, you can see in one of these would be the iron in the middle here. So the Fe is iron. That is what will bind oxygen. So you can have 1, 2, 3, 4 oxygens bound per hemoglobin. So in terms of the life cycle, the production of erythrocytes, it's going to require glucose, lipids, amino acids, iron, copper, zinc, and B vitamins. Its lifespan is about 120 days. Old cells are going to be usually removed by the liver and the spleen. They'll be broken down, reuse what it can. The non-iron part of that heme is broken down to bilirubin and biliverdin. Biliverdin, that's what causes the coloration. You know, when you get an injury and you get a bruise and you go through that lovely stage of it's kind of yellowish, kind of greenish yellow, and then it gets to the purple, and then finally the black. Well, part of that is due to when you have an injury, if there's blood that has been like just below the skin, it's been bleeding and pooling there. And then as that heme is broken down, the biliverdin is responsible for a lot of that coloring. Jaundice, which is a yellow coloration, that's caused by the bilirubin. Usually it will accumulate in the liver, and the liver breaks it down. It basically goes, the liver supplies it to the intestines, and it gets broken down. Well, if the liver is not functioning properly, it can't break down the bilirubin. Bilirubin has a yellowish color to it, and then that tends to accumulate, and it will be, you'll see the jaundice at that site. So this diagram here is just showing that in terms of the production of the red blood cells, you start with the stem cell, and you don't have to know all the steps, so just know that you start with a stem cell, it goes through that differentiation, and eventually you end up with the mature red blood cells. You've got the hemoglobin that is placed in there. When the cell breaks down, the hemoglobin is released. Here you've got your bilirubin and the biliverdin. The iron often is stored in the liver as well. It will reuse what it can, and get rid of anything it can't use. So this just cycles over and over. And so jaundice, like I said, is if the liver is supposed to be able to break down the bilirubin, if it can't because of either disease or obviously some type of abnormality in the liver, you'll start to see signs of jaundice. Jaundice is where, like I said, you get this yellowing and the skin takes on a yellowish hue. One big place you can often look at are the eyes. Look at the whites of the eyes, and that is where you'll see them turn yellow because of the bilirubin. Different disorders with the erythrocytes. There are several disorders. We'll just mention a few. If you have too few red blood cells, that's anemia. There's different types of or issues with being anemic. It can be due to blood loss. It can be due to either faulty or decreased red blood cell production, such as in sickle cell anemia, iron deficiencies, or vitamin deficiencies. Or you're excessively destroying the red blood cells. So you're destroying them faster than you're able to make new ones. And polycythemia is when you have too high of red blood cell numbers. That can sometimes be the result of dehydration. When you have a lot of red blood cells, remember they're viscous, so it increases the blood viscosity. Depending on how severe it is, if it's severe cases, you may need to be placed on blood thinners. There is a mild form of this with people who are living at a higher elevation. As you go up in elevation, the oxygen concentration decreases, so that's part of your body's natural response. Low oxygen, remember I said it's going to increase the number of red blood cells. For sickle cell anemia, for those of you who are not familiar with this, it's actually genetic. There are certain populations that at a higher risk of having it, that we just see because it's genetic in certain populations. You have your normal red blood cells here, and it actually has that sickle shape to it. And on this screen, it's very easy to notice there are several cells here that have that abnormal shape. That can make it much more difficult for the blood to easily flow through the blood vessels, especially the small diameter ones. And just so you know, there are a lot of side effects from having sickle cell anemia. It can, because the easiest way of looking at it is, you think, oh, well, it's a blood disorder. Well, you know what? All the tissues need blood. So there's this trickle-down effect where you can end up having digestive issues. You can have issues with muscles. You can have issues with the urinary system. With all the systems, eventually are going to be affected. And so it is a very, very serious disorder. The leukocytes, which are your white blood cells, their role is in with your immune system for defense and protection of you. Now, these are complete cells. They have the nucleus, they have all the organelles. They will be found in the bloodstream, but a lot of them can leave the bloodstream and go to, in the interstitial fluid, go to the site of where, say, there's an infection. Some of them are just kind of roaming around, kind of like they're on reconnaissance. You know, I'm out here patrolling. They are looking for anything that is considered foreign because they can distinguish between your cells and foreign cells. And we'll study that with the immune system. When they move out of the capillaries, that's often referred to as emigration. They can squeeze between little pores. So when you have an injured site, and we'll review this when we cover the immune system once again, but just briefly so you know what's going on here. There's an injury here. There are certain chemicals that are released that will attract the leukocytes to this area. That's how they know to come here. There's a chemical, we call it chemotaxis, positive chemotaxis. They are attracted to this area. So here in your blood vessel, you'll notice the white blood cells are larger than the red blood cells because they're a complete cell. There's different types of them. We'll get into this in just a second. But these are your white blood cells. And notice there's these little spaces between some of the cells that are able to squeeze through that. And then they will come, like I say, they're attracted to this injury site. So they're going to come over here where the chemicals are attracting them. And they're going to basically eat up any foreign debris here. So any of these bacteria, it's like, ooh, chow time. We're going to munch them down. And they also will clean up any damaged tissue. They're the cleanup crew. So any debris that's like cellular debris, tissue debris that's here, they're going to clean it up. They are attracted here because you want the white blood cells there as soon as possible because you don't want any of these bacteria to get into the bloodstream because once they're there, they can go anywhere in the body. So they're going to try to restrict the invasion just to this site. Don't let it penetrate any deeper. So they are able to leave the bloodstream. Some of them naturally will do that, like I said, kind of on patrol. Some of them do it because they're attracted to those chemicals that are released from the damaged tissue. When we look at leukocytes and the classification of them, there are 5 different types of leukocytes. And we first divide them into 2 groups, either granular or agranular. This is done simply by what they look like when you add stains to it so you can see them. You can't see them without staining them. And then you add the stain and you look at them under a microscope. What color are they? How does the nucleus look? One of the first things you're going to do is look and see, does it look like there are little granules, like somebody dumped sand in the cell? 3 of them are granular, 2 of them are not. So we look at the granular leukocytes, the neutrophils, the eosinophils, and basophils. Neutrophils, when you look at them, those granules are going to be a very light purple. You look at the nucleus. There's 2 to 5 lobes. It's not a nice little round nucleus. It has all these little lobes on it, if you will. There's 2 to 5 lobes. What do the neutrophils do? One of their main things is to carry out phagocytosis against bacteria. So if you know what the normal concentration in a sample of blood is. If somebody has a bacterial infection, you would expect the neutrophil concentration or numbers to be much higher than normal. So you know what normal is. When you do a blood sample, what your lab tech is going to be doing is looking at sample blood, and they can do several different things. They calculate how many, out of the field of view that they're looking at, how many red blood cells are there versus white blood cells. So you know what the ratio of that should be. You know about how many platelets should be there. So you look at the overall, oftentimes you count or have to estimate what's the red blood cell to white blood cell to platelet ratio. Now, if that's out of order, the other thing that you do with the leukocytes is look at what we call a differential count. You're going to look at all 5 of these types. You know what the normal range should be, and if one of them is higher than normal, that can give you an indication as to what is going on with that patient. So like I said, if you have bacterial infection, you would expect the neutrophils to be higher than normal. The eosinophils, those granules are going to look kind of a reddish-orange color. The nucleus is only going to have 2 to 3 lobes. This is seen more so if you had a parasitic worm infection, which hopefully none of you have to deal with that. I mean, hopefully none of you are ever sick, but allergies. Now, I have a lot of issues with allergies. So they'd want to look to see my eosinophils are probably right now a little bit higher than normal because it's allergy season for me. The basophils, now these will have a dark blue granules. The nucleus typically only has 2 lobes to it. Basophils tend to release histamine. It's part of the inflammatory response. They can also have higher numbers to fight parasitic infections. But they are also involved with allergies. If you have allergies, what do you usually take? Antihistamine. You're trying to counteract the release of the histamine by the basophils. Now, when I say they can do a differential count to try to figure out what's going on, here's an example of how you can, actually apply this if you're thinking, why do I ever have to know this? Here's an example of something that happened to me personally. When my son was only a month old, I had gotten really, really sick after he was born. I had a uterine infection after giving birth. When they realized what was going on, they put me on medication, obviously antibiotics, to try to treat the infection. And the timing of it was just before Christmas. So I started taking the medication, and then I had a really bad, I just suddenly got a whole lot worse. And so I was one of those annoying patients. Here it was Christmas Eve, and I had started taking the antibiotics, and I was not getting any better. I suddenly got really, really sick. Well, my concern was, you take an antibiotic, it's not going to start immediately. It's not like you take the antibiotic and 20 minutes later, hey, I'm feeling better. It takes a little bit. So I didn't know at this point, is the infection getting so much worse, it's going to take time for those antibiotics to kick in? Or am I having an allergic reaction now to the antibiotic on top of everything else? Because I had worsening symptoms once I started taking the antibiotic. So I didn't know if I was having an allergic reaction to the medication, or is it the infection that's getting worse? And so I was that annoying person who I finally tell my husband, you have to take me to, urgent care, because I don't know whether to continue taking the medicine. If I'm allergic to it, I can't continue taking it. Then we're going to have a really bad problem. So we're trying to get to urgent care. We get there 5 minutes before they close on Christmas Eve. At that time, we were living in New Mexico. I had no family. We had no family in New Mexico at all. So here we come in 5 minutes before closing on Christmas Eve with a 2.5-year-old and a newborn. And I am sick. And you could just see the look on everybody's faces like, oh my goodness, we just want to go home. And so, they're doing the usually, let's take your temperature, let's get your blood pressure, your pulse rate, oxygenation rate, all of that stuff. And I finally said, look, I know what's going on. It's either the infection is still so bad, the antibiotics that have been prescribed, they haven't had enough time yet to start working, and the infection is just getting worse, or I'm having an allergic reaction. And I know you guys all want to go home. So do a differential, take a blood sample. You only have to, and you don't need a lot of blood, you just need a small amount. So do a differential blood count. And then about that time is when they look at me and I go, what do you do? It's like, just trust me. I know what I'm talking about. Do a differential count. If the infection is getting a lot worse, but it's not allergies, then the neutrophils should be really high. But the eosinophils and basophils should be within normal range, because I wouldn't be having an allergic reaction. So these should still be low. They should still be within that normal low range. But the neutrophils, because it's an infection, and we already knew it was a bacterial infection, it should be really high. If, on the reverse, you already know I have an infection, so the neutrophil should be high regardless. If it's an allergic reaction, the eosinophil should be really high. And the basophils are going to be high as well. So really, you know these are going to be high regardless because you already know I have a bacterial infection. But if it's just the neutrophils that are high, keep taking the antibiotic. It will finally get a handle on this infection. If the eosinophils and basophils are high because I'm having an allergic reaction to the antibiotic, stop taking the antibiotic. We need to get you on something else. You can have those results in 5 minutes, and then we can all go home. And my 2.5-year-old is not sitting there crying, is Santa Claus going to find us here? And so they kind of looked at me and went, well, that makes sense. I'm telling you, I know this will, it makes sense. And I usually don't tell them what I do, but I'm like, look, I'm a microbiologist. I teach A&P. I teach microbiology. I teach this stuff. They ran the count, and they said, eosinophils and basophils are low. Your neutrophils are sky high. I said, okay, as long as I know eosinophils and basophils are low, then that means I'm not having an allergic reaction. And now I'll go home. I'll continue taking the antibiotic. It just needs more time. And everybody can go home and be happy, and we're done. So that's just an example of sometimes students say, well, I don't need to really know this. You never know when it may come into play. Take your knowledge and apply it. And sometimes it doesn't have to be some fancy, let's use a $1 million piece of equipment. Just kind of go back to your basics. And sometimes that can tell you a lot. So when you look at them, here's the neutrophils. Like I say, 2 to 5 lobes. See how you get these swellings in here? You've got those granules in there. They're kind of a light purplish color to them. Eosinophils, kind of the reddish orange. You got 2 lobes here. The basophil has the darker, it's usually like a dark blue and a couple lobes. But it looks like somebody dumped sand in there. So the agranular leukocytes, when you stain them, it doesn't look like someone dumped sand in there. There's 2 types. You have the lymphocytes. These are going to be involved with your immune response, part of your defense. They tend to have a very large nucleus. It looks like that is what most of the cell is this nucleus. There's 3 main types, the B cells, T cells, and then the NK cells, which is the natural killer cells. They do, as I said, play a role with the immune response. We have a whole chapter on that. The other type of agranular leukocytes are the monocytes. They tend to have a nucleus that's kind of a U or horseshoe-shaped nucleus. And it's kind of different. When it's in the blood stream, they're called monocytes. They will often leave the bloodstream and migrate into the interstitial fluid and, like I said, kind of be on this reconnaissance. When they leave the bloodstream, they mature into what we call the macrophages. So if it's in the bloodstream, it's monocyte. When it's out of the bloodstream, it's macrophage. And they carry out phagocytosis of both pathogens and cellular debris. So that cleanup crew that we've talked about, that's what they are. So the lymphocyte, you can see this huge nucleus, takes up almost the whole thing. And then the monocyte, so that kind of looks like a U or a horseshoe, depending on your point of reference. Leukocytes tend to have a very short lifespan. Once again, just like we said at the beginning, all the formed elements do begin production in the bone marrow, start out as a stem cell. Now, because leukocytes do have a nucleus, they can carry out mitosis versus the red blood cells that don't because they lose their nucleus. There's different disorders associated with the leukocytes. Leukopenia is low numbers of leukocytes. Leukocytosis is high numbers. Leukemia is a type of cancer with high leukocytes. Lymphoma is a type of cancer where you have high T cells and B cells in the lymph nodes. They can also be in the spleen, the liver, or in other tissues. The platelets are known as thrombocytes. What they are, you have these large cells known as megakaryocytes that will break up into small fragments from those, is what actually the platelets are. The lifespan's about 10 days. When platelets are formed, about a third of them are going to be stored in the spleen, and so they can be released from there whenever there's injury because they're involved with helping to repair damaged vessels. They're also release growth factors for healing and repair. So if you have a damaged blood vessel, the platelets that are in the blood will help with the repair, but there's also going to be a chemical signal that's released to release a lot of those stored platelets from the spleen, like, hey guys, we need backup here, come help us. And just briefly, once again, you're going to start off with the stem cell. It enlarges, makes this megakaryoblast, which turns into a megakaryocyte. And it's the fragments that break off from that are the platelets. So even though platelets are classified as a formed element, they're not really a complete cell. Hemostasis is this process that's used to seal any ruptured blood vessels. This is much more efficient the smaller the blood vessel is. If it's a very large vessel, it's going to try to repair it, but if the blood vessel is too large, you're still going to have internal bleeding, and you're going to have a surgeon go in and stitch it up in time to save you from internal bleeding. There's three main steps. There's first what we call vascular spasm, where the blood vessel itself starts to constrict. That decreases the blood flow through there to try to decrease the amount of blood loss from the injury site. You have the formation of the platelet plug and then coagulation. And this is shown here. So you have that injury that has occurred. Like I say, the vascular spasms, you're decreasing, trying to prevent massive amount of blood loss from here. Platelet plug formation. The platelets normally, essentially what happens on the inner lining of the blood vessel, which I know we haven't talked about yet, but the inner lining of the blood vessel is smooth. That reduces friction so that the blood can freely flow through the blood vessel. When you have an injury and you've cut part of that blood vessel, you are now exposing some of the fibers that are deeper in here, but they're not on the surface. Some of those fibers are kind of have a sticky nature to them, so that blood platelets will stick to them. So the platelets will stick to these fibers. Well, the fibers, and if you're thinking, why aren't they sticking to them all the time? Because the fibers are not exposed in a non-injured, nice, healthy blood vessel. They're not exposed. They only become exposed when you've had this tear or cut or whatever caused the injury here. Now the blood platelets are going to come in and they start to stick. And then you have these what's known as fibrin strands come in. They form like a mesh with all these platelets that keep sticking there, and then it's like a net that it forms around it over the injury site. And so it forms this plug, or this kind of like a clot there. And it does eventually form a clot in the coagulation step. It prevents blood flow through that injury site. But like I said, if this is a large vessel, you're going to need stitches. There's a whole series of steps that involve various chemicals, and that's what this is just showing over here. So there are disorders associated with clotting as well. Thrombocytopenia is a low platelet number. If you have low platelet numbers, that means that's going to affect your ability to form clots. You're not going to be able to form clots as easily, which means any slight injury can result in excessive bleeding. Hemophilia is an inability to produce some of those clotting factors. that in the previous slide on the right-hand side, those chemicals. Hemophilia is genetic, and if you don't produce those clotting factors, you cannot produce a clot, which once again, you can have excessive bleeding just by very small, like bumping, small injury, something that for most people would be no big deal, but for someone with hemophilia, they have to be very careful. Thrombosis is excessive clot formation due to high platelet numbers, so it's the reverse. Thrombus is a clot. Embolus is a clot that breaks free and it's in circulation, so it's moving in your circulatory system through all the blood vessels. An embolism is when that embolus or that clot becomes trapped. And the problem with that is if it becomes trapped in a blood vessel, number one, you don't want a clot to begin with, because there is concern, obviously, if you have a clot, if it breaks free and it's now moving in your circulatory system, and if it becomes trapped, it can prevent flow of blood through the vessel and the points beyond there. All tissues need constant supply of blood because they need the oxygen, they need to get rid of the CO2, they need to get the nutrients. Without any of that, they will start to die. And depending on where that blood clot becomes trapped, if it becomes trapped in the lungs, it's known as a pulmonary embolism, and it affects your ability to get rid of the CO2 and get the oxygen, and it can kill. If it's in the brain, it's a brain embolism, and it can cause parts of the brain to die. So it is a very serious thing. Blood typing. The major type of blood system is what we call the ABO blood typing. There's other ways of typing blood, but this is what we call the major. There's minor. If somebody were to be having an organ transplant, they would do a very extensive, what we call major and minor cross matching on this. With the ABO, the major cross typing, what you are looking at are antigens. These are markers that are on the surface of the red blood cell. There's two markers. There's an A marker and a B marker. So we call that the A antigen and the B antigen. That is what determines your blood type when using the ABO system. The antigen is what is on your red blood cell. The antibody is a protein that is made in response usually to an antigen. Usually an antigen is something that is foreign. In this case with blood typing, the antibody, this is what is floating around in the plasma. The antigen is what is on your red blood cell. So let's look at this first. With the ABO blood typing, you have four possibilities. An individual can be type O, type A, type AB, and so you have O, A, B, and AB. Now, a person who is type O, they do not have any antigen on the surface of the red blood cell. Floating around in their plasma is anti-A and anti-B antibodies. A person who is type A, they have the A antigen on the red blood cell. They have anti-B floating around in the plasma. A person who is type B has the B antigen and they have anti-A floating around as the antibody in their plasma. A person who is type AB, they have both the A antigen and the B antigen. They don't have the antibodies against those in their plasma. to figure out who can receive what blood types, you have to understand when you are receiving blood, you are getting the red blood cell. So you have to look at what antibodies do you have and what's on the red blood cell that you are receiving. So for instance, I know that I'm type O blood. So I don't have any antigens on my red blood cells, but I have anti-A and anti-B antibodies in my plasma. So you can always receive the type blood that you are. So I know I can get type O. I cannot receive any of the other blood types. Why? Because I would be getting the red blood cell. So I have to look at the antigen. Type A blood has an A antigen on it, but I have anti-A antibodies. This will bind. The antibody will bind to the antigen, and it will start forming this clumping precipitate, and it's not going to be good. It will kill you. This reaction can start immediately, and if it is not reversed in time, it will kill. I cannot receive somebody's blood that's type B because the B antigen will bind with the anti-B antibodies that I have. And certainly AB is not going to be a pretty picture for me. So if you're a type O, you can only receive type O blood. If you're a type A, you can receive type A, and you can receive type O because there's no antigen on those red blood cells, so there's nothing to interfere here. If you're a type B, you can get type B and type O. And because a person who is type AB blood, they don't have antibodies, they can receive all types. So everybody can get O. So O is known as a universal donor because they can give their blood to anybody. Type AB is known as a universal recipient because they can receive all types of blood. Now, there's another thing you have to take into effect, and that is what is known as the Rh factor. That is another antigen. The Rh, just FYI, it stands for rhesus because it was discovered in a rhesus monkey. In a lab, we often refer to it as D. 85% of the population is Rh positive, meaning you have that antigen. Now, this is a little different. The 15% of the population that's Rh negative, this is a case where you don't have that antigen. If you are exposed to it, you will produce antibodies against it, but only if you are exposed. So if you've never been exposed to it, it's not a problem. One of the major issues that we have, or problems with this, is if you have a mother who is Rh negative and she is pregnant and the fetus is Rh positive. If the mother has previously been exposed and has antibodies against that Rh factor, you can have severe consequences. The mother's blood does not mix with the blood of the developing fetus. It is totally separate. However, proteins can cross the placental barrier. Antibodies are proteins, so they can move from mom to the developing fetus. It's not going to do anything to mom, but the antibodies can start attacking the Rh positive, that Rh antigen of the developing fetus, with devastating results. So if they know, and they will do blood tests very early on when they know a woman is pregnant to determine her blood type, to determine if she is Rh positive or Rh negative. If she is Rh negative, they are going to watch her very closely. They will administer what is known as RhoGAM, usually around week 28. They can do it as early as week 26. What this will do is it tries to tie up any antibodies that the mother might have produced them, preventing them from passing across the placental barrier into the developing newborn. And usually right after birth, usually within 24 hours, they can go as much as 72, but they try to administer another shot of RhoGAM as soon as possible after birth to try to prevent, once again, the mother from producing any more these antibodies, tie him up. And certainly, if she already has had one pregnancy, they're going to watch really, really closely because chances are she's made antibodies. She's been exposed from the first pregnancy, so they're going to watch very closely. It can lead to what's known as hemolytic disease of the newborn. They're oftentimes, not always, but oftentimes they may have to do a blood transfusion on the newborn. It's a tough way to start life out. Just so you know, I can remember when I first was studying this, I had a professor who commented and said, it's real easy to say, well, okay, it's not a problem for those people who are Rh positive. That's 85% of the population. So you're only talking 15% of the population. And if the major issue has to do with pregnancy, you figure that 15%, half of those, let's say, are women, so you're talking about 7.5% of the population, but they have to be women of childbearing age, and it's usually with the second or subsequent pregnancy, not the first one. And he had it broken down, to, it was less than like 1% of the population that you're actually talking about where this is a problem. And it's easy to say, okay, it's just a small group, it's not that big of a deal. But if you are in that group, this can be a huge deal. A huge deal. Like I said, there are ways we know to monitor. You can try to get the program. It's not a guarantee, though. I actually had a student one time who shared with the class on her own accord that she has, at that time, had 2 children. But before she had her first child, she had 5 miscarriages. All because of this incompatibility of the blood. She was Rh negative. And even though they gave her the program, she had some additional complications with it. So if you're, just remember that whether you're talking about incompatible blood types or anything, it's that all of you guys are going into some form of healthcare. Some of you are going into nursing, some of you are going into x-ray, surgical tech, sonography, all different types of stuff. And regardless of what the issue is, for your patient, things are a big deal. When you go to do the blood typing, what happens here, and this will sometimes confuse people. For blood typing, what you are doing is you are taking a drop of the patient's blood. You are adding essentially antibodies to it to see if you get clumping or not. This is not the anti-A antibody that the patient makes. This is what a synthetic form that you are adding. So yes, I know a person with type B blood naturally makes anti-A. That's harder for you to see. You've got to run tests to see what the antibodies are that are present, some chemical tests. That's going to take a while. You need to figure out a person's blood type right now. So the quick, easy way is to, well, let's get synthetic antibody. Let's make a synthetic anti-A, anti-B, and anti-D. Remember, the D refers to the Rh factor. So determine the blood typing. If I have a drop of blood here and I have anti-A antibody, and I see this clumping like this, not like this. This is what it should normally look like, your drop of blood. That's what it initially looked like. You add this anti-A and you start getting this clumping like this. Why? Because the anti-A is binding to the A on the red blood cell, which means this person has an A marker. They have the A antigen on their red blood cell. Nothing happened here. So if you only have the A marker, that means you're type A blood. If both of these had clotted like this, they would be type AB. If nothing happened, then you'd be type O. If this was fine but clotting over here, then it would be type B. The anti-D, remember I said the RH factor in the lab, we refer to it as D. There's clotting, which means the person has the RH factor. So this individual would be type A positive, because the RH factor, that's where we get the positive and negative terminology that we use. So you look for the clumping. I usually tell students, I know oftentimes in labs, when we have in-person labs, they like to do the actual blood typing. Most universities and colleges now do not do that. We use a synthetic blood, so it reacts the same way, so you can see this type of reaction. I often get asked why we want to do that. There's different things. Part of it is safety. As an instructor, I have to watch all the Lancets, make sure people aren't reusing them or sharing them and things like that. There's also some other issues that come up. In the course of my teaching career, I have twice had this happen when we used to do our own blood typing, where a student would do, the first time it happened to me, it was a student with a very high GPA, very smart woman. I'll never forget, she was 27 years old. And she came up to me and had a slide similar to this picture and was asking for help to interpret the results. And she had it right. And so say, I don't remember exactly what blood type it was, but let's say this is what she had. So I said, yes, you have clumping with anti-A, clumping with anti-D, so this would be A positive. Your blood type is determined genetically. And we had already covered some genetics on that. So she looked at it and then she said, well, I know that my mom is type B and my dad is type O. Now, if blood type is determined genetically, if you have one parent that's type B and the other is type O, there is absolutely no way you can end up with a child who's type A, which is what she was getting at. Which I was like, well, maybe you mixed up these two. And she looked at me and said, I've already done it 3 times. And I was like, oh, well, maybe the supplies have been sitting around for a while. They may be expired. I mean, this is just a teaching lab, so we don't have, we kind of get the leftovers sometimes on stuff. And I'm trying to come up with all kinds of excuses. And she knew immediately. She said, if mom is type B and dad is type O, there's no way I can be type A. That cannot be my dad. And she was a little upset and left the lab saying she was going to go make a phone call to her mother. Yes, it's kind of funny when you tell the story years later. It was not funny at the time. It was not funny for that student essentially to find out in lab that the person she thought for 27 years was her dad was apparently not her biological dad. That has happened to me twice in the course of my teaching career. So I am perfectly happy that when we have in-person labs, we don't do blood typing with your own blood. We use synthetic blood. It reacts the same way, so you can see how it works. And I don't have to deal with some of these issues. I also had a student one time who was too scared to stick her finger. You only need 3 drops of blood. You just 1 drop, 2, and 3. Just that's all you need. And she was scared to poke her finger. And after 20, 25 minutes of staring at her finger and trying to poke it, she just raised her arm back and just flung it. She sliced her finger open. And then I had an issue of getting her to the health clinic for stitches because she had more than 3 drops. So this is the blood typing. Hopefully not traumatic. In terms of giving blood transfusions, if there is time to do blood typing, you try to give the same type. So if somebody's A positive, you give them A positive. If they're B negative, you give them B negative. If it's an emergency and you don't have time to do the blood typing, then you give the universal type, which is type O, specifically O negative. I had a friend of mine, I used to live in Oklahoma, and a good friend of mine who's a nurse was working at a hospital in Oklahoma City when the Oklahoma City bombings occurred. And they were the closest hospital, so they received most of the victims. You have people coming in, and you know that you have this disaster and multiple casualties coming in, multiple victims coming in, and there's a certain point you just hope you have enough O negative. And so when you need somebody's bleeding, you need blood transfusion right now, you don't have time to run a test, you grab O negative because everybody can get O negative. Universal recipient is type AB positive. They can receive any type of blood. So if a patient knew what they were, you could always just, if they're AB positive, give them anything. In an emergency where you don't have enough blood available, you can start to give saline that has electrolytes in it that matches what the blood plasma levels are in an effort to stabilize the volume, because the body's going to respond and try to keep the blood volume as constant as possible. Now, within the body, it adjusts where you need maybe a little more blood here, a little less blood there, but overall the volume remains fairly constant. And to stabilize that when you have excessive bleeding, the heart's going to start making adjustments, adjusting blood pressure, pulse rate, all of that to try to respond to this changing volume, to try to increase the volume. So you can give the saline that will help. It's not a cure. It can only work for so long. They are trying to come up with ways of making a synthetic blood that will work in the body that would then be able to be administered once again where you have a situation, whether it's a natural disaster or say in wartime, something where you have a lot of victims that you're trying to treat at once where you may be running out of the blood supply.